Welcome back, everyone, to another deep dive. And a special welcome if you're tuning in from the HIV-RNA Test YouTube channel. Thanks for joining us.
Great to be here.
Today, we're looking at something, well, potentially revolutionary in HIV treatment. It's a gene therapy called AGT103-T.
Yeah, and the core idea is pretty fascinating. It's about essentially training your own immune system to fight back against HIV.
So the implication being maybe no more daily meds. Is that the big picture?
That's the hope, moving away from just managing the virus day to day towards something, well, more durable.
Right, what some are starting to call a functional cure. And for our listeners, especially those who rely on resources like HIV-RNA test here in the U.S., understanding this feels crucial.
Absolutely. So our mission today is really to unpack AGT103-T, how it works, what the science actually says, the trial results so far.
And what it might mean down the line for HIV treatment, specifically here in the States.
Okay, so let's dive in. AGT103-T. It's a gene therapy developed by American Gene Technologies, or AGT.
And the fundamental concept is using the patient's own immune cells, these CD4 plus T cells, to combat HIV, right?
Exactly. Those are the cells HIV normally targets. The therapy aims to turn the tables using those very cells.
Okay, so how do they actually do it? I understand it's like a three-step process.
Pretty much. First step is collecting these CD4 plus T cells from the patient's blood. It's a process called leukapheresis, kind of like a more involved blood draw.
Okay, familiar enough. Then what happens to those cells?
Right. So step 2 happens outside the body, in the lab. The collected T cells are genetically modified.
Modified how?
They use something called a lentiviral vector. Think of it like a delivery vehicle. It inserts a tiny piece of genetic material, a microRNA or miRNA, into the T cells.
Okay, a lentiviral vector carrying miRNA. And what does that miRNA do?
It's designed specifically to block several important HIV genes. Basically, it prevents the virus from replicating inside these specific T cells. It makes them resistant to HIV infection.
So you engineer these T cells to be HIV-proof, essentially.
That's a good way to put it. Then step 3 is infusing these modified HIV-resistant cells back into the patient.
And the idea is these supercells then multiply.
Exactly. They multiply, they form a population of HIV-resistant immune cells, and the goal is for them to control the virus long-term, even without daily RT.
So the aim isn't just suppression while on meds, but enabling the body to control HIV on its own, restoring immune function to achieve maybe lasting remission.
Precisely. It's about giving the immune system the tools it needs to manage the virus autonomously.
Let's dig a bit deeper into the science then. You mentioned it's an autologous therapy.
Right. Autologous just means the cells come from the patient themselves. You take their cells out, modify them, and put them back in.
Which avoids issues with rejection, I imagine.
Exactly. It minimizes risks like graft versus host disease because the body recognizes the cells as its own.
Okay, so back to those steps. Extracting T cells. We know CD4 cells are crucial for immunity, but they're also HIV's main target.
Correct. HIV depletes these cells, which weakens the whole immune system. So step 1 is getting a good batch of these critical cells to work with.
Then the genetic engineering part. You mentioned the lentiviral vector. Is that safe? People hear viruses and get concerned.
That's a fair question. These are highly engineered vectors. They're derived from viruses, yes, but they've been modified so they can't cause disease. They're just very efficient at delivering that genetic payload, the miRNA, into the target cells.
And this miRNA tackles multiple HIV genes, not just one.
Yeah, and that's a key point. By targeting several genes, it makes it much, much harder for HIV to mutate and develop resistance to the therapy. It's like attacking the virus from multiple angles simultaneously.
A multi-pronged defense. That makes sense. And then reinfusion. These modified cells go back in.
And the expectation, based on the science, is that they'll engraft, expand, and create this durable HIV-resistant immune cell population.
Basically acting as a new line of defense.
You could think of it like upgrading your immune system software, giving it a specific update that makes it resistant to this particular malware HIV.
That's a helpful analogy. Okay, so how is this AGT103-T really different from other approaches? People might be thinking about vaccines or just their daily pills.
Good question. First off, definitely not a vaccine. Vaccines aim to prevent infection. AGT103-T is a treatment for people already living with HIV.
Okay, treatment, not prevention. And compared to daily art.
Well, daily art is incredibly effective at suppressing the virus, keeping people healthy, but you have to take it every day. If you stop, the virus usually rebounds because ART doesn't eliminate the hidden viral reservoirs.
Right.
AGT103-T is aiming for something different, a potential one-time treatment that leads to long-term control without needing those daily pills. That's the big potential shift.
And you also mentioned it's gene therapy, not gene editing. What's the distinction there?
Right, so gene editing, like CRISPR, involves making precise changes to the existing DNA sequence in the cell. Cutting and pasting, essentially. Gene therapy, in this case, means adding new genetic material, that protective miRNA gene, using the vector. It doesn't alter the cell's original DNA structure in the same way.
And why choose therapy over editing here?
Generally, at this point, adding a gene via a well-understood vector, like a lentivirus, is considered perhaps a more predictable and controlled approach compared to editing, which is newer and still carries some theoretical risks about off-target edits. So, arguably safer based on current tech.
So adding a protective function rather than trying to cut out or rewrite something.
Exactly. The bottom line is, AGT103-T isn't just suppressing the virus like RT, it's trying to fundamentally retrain the immune system to control it.
That really clarifies the difference. Now, what about progress? Has this actually been tested in people?
Yes, it has. They launched a phase 1 human trial back in 2020. This was primarily focused on safety and, seeing if the process was feasible in people living with HIV who were already on stable RT.
Yeah, what came out of that phase 1 trial? Anything significant?
Very much so. First, the big one, safety. There were no serious adverse events reported that were linked directly to the AGT103-T therapy itself. That's always hurdle number one.
Crucial. What else?
They also found that the modified T cells, the ones with the miRNA, actually persisted in the body for a good amount of time. They didn't just disappear quickly.
Which suggests it could have a lasting effect.
Exactly. Durability is key. And interestingly, they observed some reduction in the size of HIV reservoirs in several participants.
Wow, okay. Those reservoirs are the tough nut to crack with standard RDA, right?
Precisely. ART doesn't really touch them effectively, so seeing any impact there is quite encouraging.
And the most exciting part I read about was the RT interruption.
Yes, some participants under very careful medical monitoring were able to stop taking their daily RT for quite extended periods without the virus rebounding.
That's pretty remarkable, maintaining viral suppression off meds.
It really is. It's a key proof of concept result. It suggests the therapy can potentially allow the body to control HIV on its own.
Okay, so these phase 1 results sound genuinely promising. What happens next then? Phase 2?
Correct. Based on these encouraging findings, AGT is planning for phase 2 trials. These will involve more people, likely a more diverse group.
And presumably they'll be looking more closely at efficacy, like how well it works for longer periods without ART.
Exactly. Phase 2 is where you really start to gauge effectiveness in a larger population and gather more data on how long the effect might last.
Now, thinking about our listeners, especially through the HIV RNA test channel, some might be wondering, could I participate? What's the deal with trial eligibility right now?
It's important to remember clinical trials always have specific criteria. For AGT103-T, generally you need to be HIV positive, usually stable on your current RT,
Okay.
have a reasonably healthy immune system, often measured by your CD4 count. And critically, these trials are currently based in the US, so you typically need to be a US resident.
Right. So if someone listening in the U.S. thinks they might fit that profile, what should they do?
The absolute first step is talk to your HIV doctor or healthcare provider. They'll know your specific situation and can advise if any trials, this one or others, might be appropriate.
Makes sense.
You can also look for information directly from American Gene Technologies or check official trial registries like clinicaltrials.gov, but always discuss it with your doctor first.
Good advice. And just to reiterate, for anyone listening who maybe isn't sure of their status, getting tested is always step one. Knowing your status opens the door to all these options, and resources like HIVRNAtest.com offer that testing nationwide, confidentially.
Absolutely critical first step.
So looking ahead with AGT103-T, phase 2 is planned.
Yeah.
What's the ultimate dream goal here?
The ultimate goal is achieving durable RT-free remission, allowing people living with HIV to maintain an undetectable viral load and a healthy immune system without needing daily medication, potentially. Indefinite.
That would be life-changing for so many.
It really would. And if phase 2 results are strong, showing clear benefit, there's a possibility the FDA could grant it fast-track status. That's a pathway designed to speed up the review process for promising therapies targeting serious conditions. So it could potentially become available sooner rather than later, relatively speaking.
Okay, so there's a lot of hope here, but we also need to be realistic, right? How should we balance the excitement with, the current reality.
That's exactly the right approach. Reasons for excitement, solid science, really encouraging phase 1 data, a reputable company, FDA oversight, it all looks positive.
But the cautions.
Well, it's not proven as a widespread cure yet. It's still in clinical trials. Phase 2 needs to confirm the phase 1 findings in more people. And even with Fast Track, getting it approved and widely available takes time, likely years.
So promising, potentially groundbreaking, but not a guarantee and not immediate.
Precisely. But that said, the progress is undeniable. Many researchers feel we've genuinely never been closer to achieving a functional cure for HIV. The momentum is real.
So for listeners taking all this in, what are the actionable takeaways? What can people do now?
Okay, number 1, always. Get tested if you don't know your status. Early diagnosis is key. Again, HIVRNAtest.com is a great resource for finding testing across the U.S.
Crucial. And if someone is HIV positive,
Talk to your doctor, discuss your current treatment, ask about new developments, ask about clinical trials if that's something you're interested in. Be an active participant in your care.
stay informed too, I guess.
Yeah,
This field is moving fast.
absolutely. Follow the progress of AGT103-T, but also keep an eye on other approaches. There's work on things like EBT101, various mRNA strategies. It's a dynamic landscape. Staying informed is empowering.
And maybe just sharing this information.
Definitely. Spreading accurate information helps reduce stigma and keeps the community informed about the hope that science is offering. Knowledge is power here.
It really does feel like we're at maybe a turning point in how we approach HIV. AGT103-T might not be the final answer, the magic bullet, but it feels like a significant stride.
I agree. It represents a concrete step towards that future where the immune system itself can control HIV, freeing people from the need for daily medication.
The potential impact on quality of life, reducing side effects, lifting that daily burden, it's profound.
It offers immense hope. Hope for a life less defined by medication regimens and maybe less defined by the stigma that unfortunately still exists.
It certainly does. And it makes you think, doesn't it? What would a future without the need for daily HIV medication really mean? For individuals, for communities. It's something powerful to consider as this research moves forward.
Absolutely. And if you want to keep up with developments like this, please do like this video, share it, and subscribe to the HIV-RNA Test YouTube channel for ongoing updates on treatments and testing.
And one last reminder, for any HIV or other STD testing needs, check out HIVRNAtest.com for fast, confidential service available nationwide across the United States. Thanks so much for joining us for this deep dive today.
Thank you.